Electrophoresis is nothing but with the application of current, how does the separation takes place. So that is electrophoretic study. Next is susceptibility. Here we are going to see how a bacteria is going to react, how these organisms are going to react, how are they reactive to these antibiotics. So that we call it as a susceptibility study. Whether is it reactive to penicillin or is it developing an immunity to this penicillin. So those things we call it as a susceptibility study. Next is a smear, which is nothing but a sample of tissue that is spread over a slide. We say as a red blood cell smear or a blood smear commonly for malarial parasite study. Next comes specimen. What is a specimen? It's a sample that you are going to subject it to a study. That is a specimen, and that block. So when you are going to freeze it, it becomes a block, and that block you are going to cut it into various sections called as a cassette. So a specimen can be divided into various blocks and cassettes. Next comes immunoflourescence. What is to fluorescence? Fluorescence is a property to emit radiations or light, just like as we saw this radioactivity. In immunoflourescence, a dye-like substance is being added so that when an antigen-antibody complex occurs, it results in the emitting of color, say a green color or an yellow color or a red color. So with this immunofluorescence property, we are easily able to identify this antigen-antibody complexes, and thereby you can easily figure out the infectious organism that is present in the sample. There are various techniques that are also used here. One is the direct probe technique, where there will be adding substances that have a specific action on the specific target. So that is a direct probe technique. An amplified probe is that where when you are going to amplify the target, in the sense like the nucleic acid here is going to be amplified so that you can easily detect the presence of the pathogenic organism.